Bacteria reproduce asexually, which means there's no genetic exchange of information when a bacterium reproduces. Essentially, this means that one bacterium is a clone of another in a particular population. And in most bacterial populations grown in a lab, they are produced from just a single or small number of bacterium. In a natural population, there may be genetic variation within a particular population of bacteria, but they're still producing essentially identical organisms through asexual reproduction. So in order to increase the genetic diversity in a bacterial population, bacteria can exchange genes through what we call horizontal gene transfer. So horizontal gene transfer is the passing of genes between individual members of a species by non-reproductive means. This would be in contrast to what we would call vertical gene transfer, which would be parent to offspring. So throughout evolutionary history and even through or in sort of modern day settings, horizontal gene transfer is a really important way for bacteria to increase diversity within their populations. So, for example, about 17% of the E. coli DNA may have come from other bacteria based on homology with sequences compared to other bacteria. We're going to talk about 3 different mechanisms by which bacterial exchange their genetic information. But DNA doesn't necessarily have to be from bacteria to bacteria. It could also be viral transmission of DNA. It could be uptake of DNA from the environment, which could include eukaryotic DNA. And then we aren't talking about it here, but through the process of endosymbiosis, we get the transfer of mitochondrial and chloroplast DNA into what is going to evolve into the eukaryotic cell line. So evolutionarily speaking, this is a big deal. And for transmitting things like antibiotic resistance, bacterial gene transfer is still a big deal. So we are going to talk about the 3 mechanisms, but just 2 in this video. The first one is conjugation. This is where cells transfer genetic information through physical contact through a structure called a pilus. We'll also talk about transformation, which is where organisms take up free DNA that is in their environment. And then in a later video, we will talk about transduction, which is viral movement of bacterial genes from one bacterium to another. Conjugation is going to be a direct transfer, a one-way transfer of DNA from one bacterium to another. Now, this is going to be initiated by one bacterium, and the other one is sort of an unsuspecting recipient of this DNA. One of the requirements for conjugation to happen is for the donor cell to have something called a fertility factor. Now, this fertility factor is actually a set of genes that encode the information for controlling this whole process of conjugation. But the very first thing that happens is that the cell has to form a structure called a pilus. Now, a pilus is basically a tube that extends from one bacterium to another and then fuses with the second bacterium. And so it basically forms a bridge between the two bacterial cells, and cytoplasm can move back and forth between the two cells. Now, the ultimate goal here is for DNA from the donor cell to be transferred to the recipient cell. The recipient cell doesn't send DNA back. This is just a one-way transfer. And then for this to be effective and for the genes to be expressed, the recipient cell is eventually going to have to incorporate that DNA into its genome. If it does not, then those genes are useless and they aren't going to be expressed. I got a little ahead of myself. I already mentioned that This pilus is a one-way directional transfer of DNA from the donor to the recipient. And of course, the recipient doesn't send DNA backwards. The donor cell is the one that is going to have that fertility factor. So it has to have a gene that is going to initiate the formation of this pilus. And then it will transfer that DNA through to the other cell. Now, if the other cell does not have a fertility factor, we call this an F minus cell. So it just means that they are lacking that fertility factor. They may or may not be lacking it. We're going to assume in this particular example that they are lacking. And that other cell that has the fertility factor, we're going to call an F positive cell. And then that connection in between, another name for that is a sex pilus, or we can just call it a pilus for short. So the fertility factor contains a variety of genes. One is for the initiation of that tube from one to the other, but it also contains genes that are going to regulate the transfer of DNA into that recipient cell. So not only does that tube have to form, but then the DNA that is being transferred is going to have to be basically taken from one cell and given to the other. And as part of this, that transfer is going to start spooling through a single strand of the DNA that's coming through. So if this is, it could be a plasmid, it could be a chromosomal piece of DNA, but either way, we get single strand sort of unwinding and moving through that sex pilus. And then as it continues to move, as it sort of unspools from one piece of DNA, it enters the other cell. And then in that cell, the genes from the fertility factor for replication have to activate. And then it copies and produces a double-stranded piece of DNA. Because double-stranded DNA, that's the DS, double-stranded DNA there, because double-stranded DNA is far more stable. We can't have single-stranded DNA hanging around in our cell. We have to have that copied based on complementary base pairing. And then there are a couple of options here. One is that this new piece of DNA could be integrated into the genome. And that is going to occur by recombination or crossing over. Or that piece of DNA will just be maintained. So if it is a plasmid, that plasmid DNA can be maintained on its own. In a previous slide, I think it was the previous one, I forgot to point out the integration into the genome. But if you do go back one slide, you'll see the illustration with the crossing over and recombination into the chromosomal piece of DNA. So if the receiving cell gets the full set of fertility factor, then that cell has been converted from an F negative to an F positive cell, which means now that new cell can transfer DNA as well. So in our previous slide, it showed the piece of DNA going in and then integration in the genome. In this case, the illustration is that we have a full F factor on its own plasmid, and now this cell is maintaining it. In some instances, that F factor can get transferred into the genome, the genomic DNA of the recipient cell. When this happens, we call this high frequency recombination, or HFR for short. So if we get recombination, and there's our crossing over event that is going to combine this plasmid with the F factor into the bacterial chromosome, then it becomes part of the genomic DNA of the bacterium. And if this happens, then this F factor is going to be passed to offspring as well. But also what's important here now is that this bacterium can transfer their chromosome, their genomic chromosome. So it's not just the plasmid that's being passed now. Now they could potentially pass genomic genes between themselves and another species of bacteria. If the if the fertility factor is advantageous, or the genes on the plasmid with the fertility factor are advantageous, then the cell is going to maintain it, whether it's a plasmid or a bacterial genomic chromosome. So basically we've got some sort of selection pressure here. If the chromosome is not advantageous, whatever genes are on there, the fertility factor, the other genes, maybe their antibiotic resistance, something that provides an advantage, then that plasmid or set of genes inside the bacterial chromosome will be maintained. If it's not, then it's metabolically expensive for bacterium to maintain this DNA, and so they could potentially stop maintaining the plasmid or stop maintaining the genes in that plasmid. Our textbook doesn't go into too much detail on how the HFR could be lost, but basically it's the same idea. The HFR plasmid can potentially pop in and out. We learned that this is called an episome in our last video. These are plasmids, pieces of DNA that can pop in and out of the genome. So an HFR fragment could be lost that way, or it could simply not be maintained. If the F factor on a plasmid is not located with some advantageous genes, then that little plasmid might be lost. And that's true for any plasmid, not just one with a fertility factor. So the advantage of this is that it is one of the ways that bacteria can increase their genetic diversity. Again, they're reproducing asexually, and so if we don't have a way to introduce new genetic information, then they're not going to have it. Keep in mind that these cells may also be acquiring new information through mutation, but this is the only way that they're going to get full-blown new genes, especially genes that are advantageous, as opposed to evolution, slow evolution of genes in a particular organism. So conjugation is a natural way that genes are transferred. One common example is antibiotic resistance. So we say that the specific plasmids that contain antibiotic resistance are R plasmids or resistance plasmids, and these are a natural adaptation of bacteria. So we have isolated antibiotics from a variety of natural sources, some from other bacteria, because bacteria will produce antibiotics against other bacteria, or fungi will also do this. So for example, penicillin comes from a fungus that produces an antibiotic against bacteria, and we've isolated that, and now we use that. So just like viruses and bacteria have kind of been locked in this evolutionary arms race, bacteria and antibiotics and the organisms that produce those antibiotics have also developed strategies for one another, and one of them is a resistance plasmid. Resistance plasmids have become a problem, though, as we have started to use antibiotics more commonly, and in a healthcare setting where you're using a lot of antibiotics, resistance can develop really quickly. And if individuals are not careful, they can transfer that antibiotic resistance to other bacteria. The next method we're going to discuss is transformation. This is where a bacterium takes up free DNA from the environment. This is, of course, a natural process. It is one of the mechanisms that have evolved, but it is also one of the mechanisms that biologists have sort of commandeered for our own purpose. So transformation is a really common biotech technique. And it is the most common one that we use to get foreign DNA into a bacterial cell. And the reason for this is that it's generally kind of a mechanism that we can control. So if we were trying to use conjugation, we would have to induce the conjugation genes, and we would have to hope that they're connected long enough to get the entire chromosome through, which may or may not happen. So transformation is an easier way for us to get DNA into the cell. So what you need for this is free DNA. You need specific environmental conditions, which you can sort of set up in a laboratory setting. And then eventually you need integration into the genome, unless you are transforming a plasmid, which is another option. In a natural setting, you may or may not be transforming a plasmid. Usually transformation is for small pieces of DNA, but under the right conditions, you can get a cell to uptake a plasmid. In a natural environment, free DNA is probably going to come from another bacterium or a eukaryotic cell that has burst and released its DNA into the environment. And then if another bacterial cell encounters it and the conditions are right, then it will take up that piece of DNA. In order for those small pieces of DNA to be expressed, they also have to be integrated into the genome. So recombination has to take place between that fragment and a piece of genomic DNA. In order for a cell to actually take up DNA, whether it's in a laboratory setting or in an environmental setting, the cell has to be called what we would call competent. So competent means that the cell is sort of ready and able to take up DNA from the environment. And this is what makes this technique so attractive for the environment. There are several ways that we can sort of force our cells to be competent. One of the most common ways is by heat shock. Basically what you're doing is kind of relaxing the membrane and then sort of tricking the cell into wanting to take this DNA in. Our textbook doesn't go into it, but in order for the cell to take up a piece of DNA, it actually has to have a little protein on the side, and this is just a random sketch of one, a little protein on the edges of its cell wall and plasma membrane that is going to open up and actually pull in that DNA. And so you can force it to do that through heat shock, through electrical charge, or chemical application. Very often this is chemical application is adding a salt solution. So you might even do multiples of these to just sort of really make that cell ready and willing to take up the DNA. So basically what you're doing is sort of triggering the cell to let its guard down and bring that transformed DNA into the cell. So just a couple of quick vocabulary words here. A cell that has been transformed is called a transformant. If you are inserting multiple genes, you say that those genes were co-transformed. You don't have to transform with a plasmid. You could transform with a small piece of DNA. However, a plasmid is going to be a little bit more stable. But in this image here, it shows a fragment of DNA coming in and being integrated into the genome. So we see this double-stranded piece of DNA come in, and then as it enters, the other strand is hydrolyzed, which means broken down. We build biological molecules through a process of dehydration, and we break it down through hydrolysis reactions. So that's what's happening there. And then in order for this to be integrated, it has to be brought in by crossing over. And since it's single-stranded, that crossing over has to happen between one side of that genomic chromosome. And so then any remainder that is excised during that crossing over is going to be broken down and degraded. And then once this cell divides, so when this cell divides by a process of binary fission, it works just like in a eukaryotic cell. The bacterial chromosome is going to be copied. So one strand of the original strands, in this case it's the inside strand, one strand is going to be copied. There's no integrated piece of DNA, but the other one, the outside strand, has that gene on it. And so when that one's copied, that new chromosome is going to have that whole gene of interest. Our last mechanism of horizontal gene transfer in bacteria is transduction. This is where a virus infects a cell, picks up a piece of the host cell DNA by accident and packages it into its viral particle and then inserts it into the next cell when it infects the next cell. So we're going to talk a little bit more about this mechanism after we talk about viruses, and so we will talk about it in our virus video.